Hi, my name is Kate Megquier, and I'm a veterinarian and postdoc at the Broad Institute in the Carlson Lab. And I'm really excited today to share with you some of our work developing blood biopsy in dogs and how a new canine exome capture that we designed with Twist is enabling our work. First, I just want to give a little bit of background on what blood biopsy is. Blood biopsy is actually a really exciting new technique that allows us to sequence tumor DNA directly from the bloodstream. Over the course of normal cell turnover, DNA is released into the plasma, and it circulates for a short time in the plasma as cell-free DNA. In a cancer patient, some of this DNA will be from the tumor, and this gives us the opportunity to sequence tumor DNA from a non-invasive blood draw. And this technique actually opens up a lot of exciting opportunities in longitudinal patient monitoring, as well as the possibility for screening tests for cancer. So over the past few years, there have been a number of really exciting studies looking at human cancer patients and the applications of blood biopsy. And it's been found that blood biopsy is really useful for finding targetable mutations and matching patients to targeted therapies. It's also useful as a measure of minimal residual disease in patients and can be used to identify relapse before it's clinically apparent on imaging. In addition, it's been used to identify emerging resistance mutations, which could potentially be targeted in rescue therapy. And so there are many really exciting applications for this, but at the moment, only a few of them have made it into the human clinics, notably single gene and small gene panels for matching patients to targeted therapies. These are like the Foundation 1 and Guardant 360 CDX panels that have been approved by the FDA. But currently, blood biopsy is limited in its application in the clinics, mostly from a lack of standardization of technique, both pre-analytical factors and how the assays are carried out. We really don't understand yet all of the factors that might impact the diagnostic quality of a sample. In addition, some of these techniques lack sensitivity for detecting early stage disease in diagnostics. And there are potential confounders for using this technique as a diagnostic, including clonal hematopoiesis mutations potentially being mistaken for tumor mutations. So we felt that that dogs with spontaneous cancer would actually be an ideal population to really develop and advance this technique, both for veterinary and for human medicine. So why do we study cancer in dogs? Well, dogs suffer many of the same cancers that people do, and they often get the same treatments. So they'll get chemotherapy, radiation therapy, immunotherapy, and even targeted therapies nowadays. But even though they do get the same cancers that we do, the incidence of these cancers is very different. Here in these simplified pie charts, you can see on the left that humans, the majority of the time, are getting carcinomas, whereas in dogs, cancers that are more rare in humans, like sarcomas, are much more common. And this gives us the opportunity to potentially study some of these rare cancers in humans in a much larger population if we look at them in dogs. An example of this is angiosarcoma. This is a cancer that's very rare in people, with only about 100 cases per year in the US, whereas canine hemangiosarcoma, which is the analogous cancer in dogs, is very, very common, and we have over 100,000 cases per year in the US. Dogs also share our environment, so they have the same environmental exposures that we do, and they have the potential to act as environmental sentinels, identifying exposures that could lead to cancer in both species. At this point, there have been some studies um, that have hinted at exposures that may lead to cancer in dogs, including tobacco smoke and lawn pesticides. Now, even though dogs receive very similar treatments to humans, there are no gold standard therapies in the veterinary clinic. And so the therapies that they're receiving are much more flexible. In addition, they have a much more rapid clinical course of disease. For example, the median survival time for dogs with lymphoma or osteosarcoma is approximately a year, whereas in humans with those cancer types, the 5-year survival rate is more like 70%. And this enables clinical trials in dogs to be carried out much more rapidly. So we really felt that this was an ideal population in order to develop blood biopsy. We worked with Victor Adelsteenson at the Broad Institute to adapt the blood biopsy technique that he developed for use in dogs. And this involves taking a blood sample and isolating the plasma, extracting cell-free DNA from that plasma, and then performing ultra-low pass whole genome sequencing at a depth of about 0.1x across the genome. We then calculate the read depth across large windows of the genome. And this information is given to the iCore-CNA algorithm, which uses the hidden Markov model to estimate the tumor fraction that's most likely present in that sample. If the tumor fraction is above 10%, then there's enough DNA from the tumor to proceed to deeper sequencing, including whole exome sequencing, which is what the new exome will enable us to do. So first, we wanted to see whether this technique was even feasible in dogs. We did a pilot study looking at plasma samples from 27 dogs. 6 of these were prospectively collected, and 21 of them were small banked samples from 0.5 to 1 milliliter. And we found that we were able to isolate cell-free DNA from all of these samples, and that the amount of DNA and tumor fraction really varied over the different cancer types. This is an example of the iCore-CNA output for a dog with hemangiosarcoma from this cohort. Here on the y-axis, you have the log2 copy number ratio, and along the x-axis, you have all of the chromosomes. Gains are in red and losses are in green. And so you can see across the genome here that we have a number of copy number gains and losses in this tumor. And up at the top, the algorithm has decided that this most likely has a tumor fraction of 52%. This is an example of a lymphoma from the same cohort. And here again, you can see a number of copy number aberrations. And the tool has decided that the tumor fraction that's most likely is 82%. And this was the highest of any of the tumors in our pilot cohort. So having demonstrated that this technique was actually feasible in dogs, we next wanted to know whether or not the tumor and plasma samples were concordant in the somatic mutations that were present. So for this, we enrolled a cohort of 24 dogs with osteosarcoma, where tumor normal sequencing had already been done by our collaborator, Heather Gardner. We had paired plasma samples for each of these dogs, and so we isolated cell-free DNA and performed ultra-low pass whole genome sequencing. Then the tumor sample and normal sample were run through iCore-CNA as pair, and the cell-free DNA sample and normal sample were also run through iCore-CNA as a pair. We then compared the outputs. And overall, we saw a good level of concordance. This is an example of our most concordant tumor plasma pair with the plasma at the top and the tumor on the bottom. And you can see that that the copy number changes across the genome are really highly similar between the two samples. We also performed a Spearman correlation of the log2 copy number ratios between the plasma and the tumor sample. And this was our most highly correlated sample and had a row coefficient of 0.96. Across all 24 samples, the row coefficient was about 0.63, which is a little bit lower than was reported in the Adelsteenson et al. 2017 paper where they looked at metastatic breast and prostate cancers. But we did note that the correlation was somewhat dependent on the plasma tumor concentration. And so when we limited our correlation to the 13 samples where the tumor fraction was above 10% in the plasma, our row coefficient improved to 0.74, which was more on par with what was seen in the human study. Overall, we felt that this was a good level of concordance, and we're really excited to be able to now move on to calling simple somatic variants using the exome capture in order to more deeply investigate the concordance between tumor and plasma sample. Now, next, we were really interested in looking at clinical applications of blood biopsy in dogs. So we first looked at whether or not cell-free DNA would be a biomarker of treatment response over time. We enrolled a cohort of 10 dogs with lymphoma. These dogs had been treated with doxorubicin and a novel agent as part of a clinical trial. And we had longitudinal plasma samples from these dogs. The first was from day 0, where they had their first treatment, then one from day 7, where the dogs were in remission, and finally, a sample from a third time point that was either relapse or just the latest visit for dogs who were still in remission. And we found that both the tumor fraction and the concentration of cell-free DNA followed the disease course quite well in these dogs. This is an example of one of the dogs in the cohort, and you can see the iCourse DNA plots for each of the 3 time points. And you see at the top that there's a copy number aberration on chromosome 13, and they have a tumor fraction of 72%. At the second time point, when the dog is in remission, the copy number change has gone away and the tumor fraction's gone down to 0. Then when the dog has relapsed, the same copy number change has come back and the tumor fraction is back up to 34%. And overall, this held true in many of the cases in this cohort. Here I'm plotting the tumor fraction along the y-axis and each of the 3 time points along the x-axis for each dog in the cohort. And what you can see is that for the most part, the dogs followed this pattern where often at the first time point, they had a higher tumor fraction. At the second time point, that dropped to 0 when they were in remission. And then the dogs who relapsed had the tumor fraction rise again, whereas dogs who were still in remission remained down at 0. We did have 2 dogs, dog 21 and 28, that had 0 tumor fraction across all 3 time points, and one dog, dog 22, that remained having a low tumor fraction at the 3rd time point, even though he had clinically relapsed. And so we're looking into whether there are any clinical variables that might be associated with those differences. We also found that the DNA concentration over time matched the clinical course as well, with having a higher concentration at the first time point, and then that dropping low during the remission. It's difficult to see here with these axes. Everyone's plotted on the same axis. So here I've scaled each of the concentration on the y-axis just to that dog. And so that sort of exaggerates the change, but you can see the pattern better. And so you can definitely see that the dogs tended to have a higher concentration at that first time point. It drops low for remission, and then the relapse dogs, it tends to be coming up. And the dogs who are still in remission, it tends to stay low. Although there were cases like dog 29 that were still in remission, but that cell-free DNA concentration was coming up. And so we're really interested to look and do deeper sequencing here and see if we can identify minimal residual disease in these dogs and see whether or not any of this is predictive of their clinical course. In addition, we're excited to use the exome capture to do sequencing of each of these time points and see if we can identify emerging resistance mutations that might be targetable in these dogs. So for these applications and more, we decided to design a new exome capture. And for this, we worked with Twist. The exome capture will really enable us to do simple somatic variant calling, as well as looking at more detailed copy number changes, and even looking at some structural variants that happen to fall within our regions. For our work, we're really excited to use this to investigate the concordance between the tumor and the cell-free DNA in more detail. We'd like to be able to credential the blood biopsy as a proxy for the tumor biopsy in this way. And we're really excited about the identification of resistance mutations and matching patients to targeted therapy using this technique. So in order to design the exome, we first looked at the canine gene annotation. So we used the Ensemble Canine Gene Annotation version 99. And we went and took the regions defined by the coding exons. And so this encompassed 20,257 genes, and it covered about 35.7 megabases of sequence. That was sort of our base. We next looked at a human exome capture to see which regions were targeted there. And we took regions of interest from the human capture and lifted them over from HG19 to the CanFam 3.1 genome. We were successful in lifting over about 4.1 megabases of sequence, and that partially overlapped with the canine sequence that was already in our design. We also targeted regions of interest specifically for cancer studies. And so that included some gene promoters and UTRs, as well as some commonly mutated regions known from the human cancer literature. Twist then took our design and looked at the targeted genes and actually added additional tiling at 2x and 4x for regions that were known to be difficult to capture in the human core exome. So regions that might have a higher mutation rate, for example. And so we added that for anything that was known to be hard to capture in humans. The final design actually targeted 36.6 megabases of sequence, and the probe footprint ended up being about 40.5 megabases. And now next, we were really excited to see how well the new exome capture performed. And so we did a pilot study. We did a study of 12 cell-free DNA libraries that had tumor fraction above 10%. Exome capture was carried out by the Broad Genomics platform, and then they performed sequencing on a HiSeq-X with 151 base pair paired reads, and they had a target depth of 150x. What we saw was that overall target coverage was excellent. We had a mean target coverage of 236x. And here in this histogram, you can see the coverage along the x-axis and then how many targets in each bin reached that coverage. And so for the most part, we thought that this looked really good, but we did have this unusual tail at 0 of about 1,200 targets that had no coverage. And so we investigated that further, and we found that these regions actually don't map uniquely, for the most part, to the CanFam3 genome. And so they receive a mapping score of 0. We did a little bit more investigation, and we found that many of these regions actually do map uniquely in the new CanFam4 genome. And so for now, we're leaving them in place because we think that they may be helpful for future versions of the exome capture. Now, Twist also performed an analysis using some of our pilot samples, and they found that the performance was really comparable to the human core exome. They looked at 3 of the pilot samples, and they downsampled them to 20 million reads for normalization. The percent on target was really excellent in our study. We have the 3 samples here that you see in the plot. And our percents on target were from 77.8 to 79.5%. And the expected percent on target was actually just above 60% based on the human capture. And so those were really good numbers. In addition, the fold enrichment across all 3 samples was from 41.1 to 42%, with the expected fold enrichment being 40 to 44% based on the human exome. So overall, we saw really good metrics with our capture, and we think it performed really well. We're excited to move on to some downstream applications, including calling variants, simple somatic variants with our data. So in conclusion, blood biopsy is feasible in canine cancer patients. Dogs are a promising model for developing this technology. And tumor fraction tracks with disease burden and can be used for monitoring patients. In addition, we think that the new canine exome capture will enable important comparative studies, including our blood biopsy work in the future. And with that, I just want to acknowledge everyone who's been involved in this big collaborative project, especially my advisors, Eleanor and Cheryl, and everybody at the Broad, Tufts, and Twist, including our funding agencies, and of course the dogs and their owners that participated in our studies, and veterinarians that took samples. And with that, I'd be happy to take any questions during the Q&A.